Together with Doctors Locascio and Tovmasian, we'll give you a brief overview of our recently published study in JCI Insight. Histone deacylases have emerged as promising targets for the treatment of brain cancer. However, to date, most HDAC inhibitors tested so far have failed to provide clinical benefit in brain cancer patients. The key challenges in the clinical translation of these agents is the inability of these drugs to effectively penetrate the blood brain barrier and also the broad spectrum nature of these inhibitors. In this study, we performed detailed assessment of neuropharmacokinetics, pharmacodynamics, and radiation sensitization properties of a second generation HDAC inhibitor, cusinostat, in glioblastoma preclinical models.
So cuisinostat is a second-generation HDAC inhibitor that harbors remarkable selectivity towards class 1 HDACs. As you'll see in this dose-response curve, the cellular IC50 for cuisinostat in all cell lines tested was in the very low nanomolar range. Treatment with cuisinostat at the IC50 concentration, as demonstrated in this immunofluorescence staining, induced significant inhibition of proliferation and an increase in programmed cell death. Because HDAC inhibitors have been described to be radiosensitizers in other cancer models, we investigated whether cuisinostat could also radiosensitize glioma stem cells, which is indeed what we found. We confirmed that this effect was synergistic and at the lowest levels of drug and radiation.
It was very intriguing for us to find that there is interspecies differences in the stability of cuisinostat. Due to the presence of various esterases in rodent blood, this class of hydroxamic acid-based compounds display poor stability and high plasma clearance. We found that cuisinostat exhibits significant instability in mouse plasma. We also observed that cuisinostat degradation happens in mouse brain, which is most likely related to the enzymes that are present in the mouse blood. Surprisingly, the drug was completely stable in human plasma and brain, which is due to the absence of the specific esterases in human matrices. To study cuisinostat levels in an orthotopic patient-derived model of glioblastoma, we assessed the drug levels after short-term cuisinostat treatment. We have observed that cuisinostat accumulates much more in tumor tissue as compared to the contralateral brain tissue. Unbound cuisinostat levels in tumors were up to 600-fold above the biochemical IC50 for HDAC1 inhibition. Importantly, although cuisinostat levels in contralateral brain tissue were significantly lower, they were still above 10-fold higher than biochemical IC50 for HDAC1.
With these results in mind, we had the confidence to perform a survival study. As you can see from this Kaplan-Meier survival curve, while cuisinostat alone was not enough to induce sufficient prolongation in survival, we see that when cuisinostat is combined with radiation therapy, the mice live significantly longer than either monotherapy arm alone. So when we look at gene ontology terms that are enriched in either combination-treated mice, radiation-treated mice, and cuisinostat alone-treated mice, you can see that in the combinatorial cohort, we see massive upregulation of gene ontology terms that relate to neuronal function, neuronal identity, and synaptic activity, indicating that the tumor cells may be shifting towards a neuronal-like cell fate in response to combinatorial cuisinostat plus radiation treatment, which may indicate a potential shift in cell identity or an adaptation response to treatment. So to conclude the general findings of our study, we found that cuisinostat, a second-generation HDAC inhibitor that has very high affinity towards class 1 HDACs, is a brain blood-brain barrier penetrant molecule, and that the amounts of free drug available within the tumor tissue is sufficient to inhibit the functions of HDAC, class 1 HDACs, and in doing so, inducing histone hyperacetylation, which for mechanisms we still need to investigate, leads to increased levels of DNA damage. When you add radiation therapy on top of cuisinostat treatment, this effect is highly synergistic. Cuisinostat radiosensitizes glioblastoma tumor cells to radiation therapy. While they are sensitive to this treatment, are still able to evade therapy. Some tumor cells die, but also others shift their identity, and future steps will tackle this further.
Our preclinical results with cuisinostat are promising and have broader implications for the cancer community. But to ensure that the drug behaves similarly in humans, the obvious next step would be to evaluate it via a phase 01 trigger trial in brain cancer patients.